Okay, in this chapter, we're going to dive a little bit more deeply into the cell biology of microbes, which will include prokaryotic bacteria as well as eukaryotes. We're going to talk about the architecture of both the prokaryotic and eukaryotic cells. So, we're going to walk you through what's inside the typical cell, what it looks like, and what it does. Okay. Um, we'll also talk about some aspects that are unique to bacteria, such as the gram-negative and gram-positive cell wall and the components. And then we'll describe the biochemical composition of a bacterial cell. Now, one of the things we have to appreciate about single-celled life on planet Earth is that it comes in really three flavors. Okay, two of the flavors are prokaryotic single-celled organisms, and then the third are eukaryotic single-celled organisms. Okay, um, prokaryotes get their name from the fact that they don't have a membrane-bound nucleus, so their genetic material (DNA) hangs from a location inside the cell called the nucleoid. Okay, and it's basically floating in the cytoplasm attached to the inside of the cell membrane. Okay, and this includes the bacteria and the archaea. All right. Eukaryotic cells are much different. They are a more complex cellular architecture with both membrane bound and non membrane bound organelles. Okay. Now, there are also organelles in the prokaryotic cell, but there's fewer of them. The reason for the organelles in the eukaryotic cell is to isolate sets of chemical reactions that work most effectively in a confined space and to separate chemical reactions that would interfere with each other so that they can take place effectively. Okay? If we look here at a typical prokaryotic cell, we see an envelope, which is a cell membrane that contains embedded proteins for structure and transport. The cell membrane is supported by the cell wall. In a gram-negative cell, the cell wall is coated by an outer membrane whose sugar chain extensions protect the cell from attack by the immune system or by predators. So a lot of pathogenic bacteria are going to be what we call gram-negative. The rotary motor of the flagella is attached to the membrane. The purpose of the flagella is to move the bacterial cell from one location to another using ATP for power. The cytoplasm contains molecules of messenger RNA and extends out of the nucleoid region to the cytoplasm, which is rich in ribosomes. Okay, so again, it's all sort of floating there um, in one compartment. The ribosome's purpose is to build protein using the information present in messenger RNA. And we talked earlier about the fact that um, that process is called translation. And in bacteria, indeed in all prokaryotes, transcription and translation happen at the same time. Transcription is the transfer of information from DNA to RNA through the production of a messenger RNA molecule, and translation is the use of the information in the messenger RNA to build a protein. Okay. Um, these proteins are folded into their active form in some cases using other proteins that are called chaperones. Right. While the nucleoid is just the chromosomal DNA wrapped around binding proteins, reproduction by DNA polymerase and transcription by RNA polymerase occur at the same time within the nucleoid. Okay. Um, Again, as does translation. So you can see here some of the different components of a bacterial cell, the percentage of total weight, number of molecules per cell, and the number of different types of molecules. Okay. The bacterial cell is rather small. Okay, it's much smaller than a typical eukaryotic cell. It is approximately the size of a mitochondria. Okay. We'll talk about the functions of the cell membrane and talk about how nutrients move across the membrane and when energy is involved. Okay? So we noted earlier that the first barrier between the inside and the outside of the cell is the plasma membrane, and we talked earlier about the fact that it is a fluid mosaic, meaning it's composed of phospholipids primarily, but it will also contain integral and peripheral proteins, as well as the occasional carbohydrate, and um, will also, in some cases, have cholesterol in it. Okay. The purpose, again, of the phospholipid bilayer is to provide a semi-permeable barrier so that certain things can pass through this barrier and other things can't, and the cell determines what can pass through and when. And it also helps to keep the inner contents of the cell in one place, right, and isolate them from the external environment. So it basically holds everything together as a functional system in a discrete, confined place. The integral proteins go basically from one side of the membrane to the other, and they can serve as gates, tunnels, or channels, or pumps, okay, or receptors. Would be just a few examples of some of the roles that they can carry out. And it's called fluid mosaic because, again, it is made up of more than one type of molecule, with the proteins interspersed amongst the phospholipids, along with the interspersal of cholesterol, if there's any there, and the dispersal of the occasional carbohydrate. So what does the bacterial membrane do? It's a support structure that protrudes from the cell. It features, in some cases, flagella, and in other cases, pili. Okay, the flagella is a structure designed for movement of the cell from one place to another using ATP for power, while pili are basically hollow pipes that extend from one bacterial cell to the other for the purposes of transferring genetic information. The membrane also serves a signaling and communication role, as it has receptors on its surface that bind to ligands, which are chemicals in the environment, and those can trigger a series of chemical reactions inside the cell called a cascade that allows the cell to adapt to this new environment it finds itself in as the result of these chemical signals. It exports toxins and virulence factors. Toxins are basically molecules that injure or damage components outside the bacterial cell. In the case of a pathogen, that might include the host. Okay, and virulence factors, which are simply features of the outside of the cell membrane, that allow it to do things like attach and colonize particular tissue types. Okay, or in some cases evade destruction by the immune system. There's all sorts of virulence factors that aid in the survival of the pathogen over its destruction by our white blood cells. It's important in the transport of substances in and out of the cell across the membrane, and it establishes a concentration gradient for energy transfer. Next, we're going to talk about cell wall structure and talk about the gram-positive and gram-negative cell wall, as well as the periplasm and the lipopolysaccharide layer. Okay? So if we look at a typical bacterial cell, the entire cell wall from E. coli is made up of glycan chains, which are parallel rings of carbohydrate linked together by peptides, as shown again here by the arrows. Right? The space between the links is open, and large molecules can get through. Okay? So you might then wonder, well, then what's the purpose of the cell wall? And it's to provide protection against osmotic shock. If the cell were to enter a an environment in which there was very few solutes in the water, okay, like let's say pure water, all right, then what would happen is that water would move into the cell quicker than it moves out, and the cell would swell up. Well, if the cell didn't have a cell wall, it could rupture and die. Whereas if it has a cell wall, it'll simply expand to the point where it reaches the cell wall, and then after that, the, the, the liquid that goes into the cell is going to be equal to the rate at which the liquid goes out, and the cell will quit changing shape. Okay. The same is true of a hypertonic solution, where the cell would normally shrivel up and die because of the presence of the cell wall. It's tougher for that water to get out of the bacterial cell. Okay, so we'll see there's several layers to the bacterial outer covering that all have their own unique role. This is an example of carbohydrate metabolism. A disaccharide unit of glycan was attached to 4 to 6 amino acids in this particular cell wall layer. The peptide bonds are with N-acetylmuramic acid, and on this peptide, the extra amino group of the M-diaminopimelic can cross-link to the carboxyl terminus of the neighboring peptide. The addition of D-alanine to the peptide is blocked by vancomycin, and the cross-bridge formation that transports the transpeptidase is blocked by penicillin. Okay, so these are two effective ways to go after organisms that have this particular type of outer barrier. Okay, and the simple reason that these attacks work is because they're based on essentially the unique properties of the cell wall. Okay, so these drugs, antibiotics, go after certain steps that build this wall up. They weaken or block those steps, and the result of not having a functional cell wall is that the bacterial cell is more prone to death by osmotic shock. Okay. Bacterial cell wall that's gram-positive is going to have a thick cell wall with multiple peptidoglycan layers. Examples include Bacillus anthracis and Strep pyogenes. Gram-negative cell wall has a thin cell wall, a single layer of peptidoglycan, and is enclosed by an outer membrane. Examples include E. coli and Pseudomonas aeruginosa. Okay? The gram-positive cells can have up to 40 layers of peptidoglycan reinforced by teichoic acid threaded through the layers. The teichoic acids are negatively charged, and that helps them retain gram stain. So these gram-positive cells are going to look at the end of the procedure as purple shapes, okay, because they retain the crystal violet. Okay? Teichoic acid can also help pathogens attach to host cells. Okay. The additional S layer can be found in gram-positive cells, and it's made of proteins and glycoproteins. The S layer subunits contain pores big enough to admit a wide range of molecules, and it's an important virulence factor for a lot of pathogens, okay, because it can, among other things, either provide for the presence of a toxin, or it can allow the bacterial cell to attach to a tissue and colonize it, okay. So you can see here, basically, you've got 3 layers. You've got the membrane, you've got the gram-positive cell wall, then you have the S layer, okay, and then you have glycosyl chains attached, okay. So 1, 2, 3, 4 components here, okay. Gram-negative cells only have 1 to 2 peptidoglycan layers. The extra layer, which is external to the cell wall, is called the outer membrane, and it helps with defense abilities and toxinogenic properties, and we find those in bacteria such as Salmonella typhimurium and enterohemorrhagic E. coli, Escherichia coli. Between the outer membrane and the plasma membrane is a space called the periplasm, which contains distinct proteins not found in the cytoplasm that protect the cell from dangerous oxidants. Okay, so what's interesting about this setup, okay, is that because of the difference in the layers present chemically, um, the cell has more protection against toxic stuff getting in and damaging the way the cell can operate. All right. In addition to the fact that it can, because of its structure, house um, what we would call endotoxins, which are toxins that are physically attached to the uh, the outer cell membrane. Okay. Lipopolysaccharide is sometimes called endotoxin. Um, this is a cell component that's harmless as long as the pathogen is intact, but when it's released by a lysed cell, endotoxin overstimulates host defense and causes endotoxic shock. Symptoms of shock are low blood pressure and fever. Antibiotic treatment may cause a reaction that kills the patient. We have to be very careful here. LPS has three parts: lipid A, core polysaccharide, and the O antigen. Lipid A has a short fatty acid chain that can be branched and is attached to the core polysaccharide, which extends out of the cell, while core polysaccharide extends to the O antigen and forms a layer to help the bacteria resist phagocytosis. The outermost layer of the gram-negative bacteria is a waxy coating that's extremely hydrophobic and retards phagocytosis by lymphocytes. The waxy coating excludes antibiotics and offers exceptional protection from host defense. The waxy coating is also going to retard the uptake of nutrients, and as a result, this organism is going to grow more slowly. Okay, so this is a special type of cell wall known as the acid-fast cell wall. Okay, so this is unique from either gram-positive or gram-negative microbes. Mycolic acids make up part of the unusual envelope, and this can be stained using the acid-fast procedure, but the gram stain will not cause these microbes to show up. Mycolic acids are linked to arabinogalactan chains, and the antibiotic ethambutol inhibits arabaminogalactan biosynthesis. Okay, galactan biosynthesis. So, again, by blocking a chemical reaction or a series of chemical reactions unique to this particular microbe, we can develop antibiotics that target those processes while leaving our own cells alone, and that can allow us to recover from the disease more effectively. Okay? Moleculites are just bacteria without a true cell wall. They include mycoplasma and ureaplasma. Next, we're going to describe how DNA is organized within the bacterial cell and talk about how DNA replication is coordinated with cell growth and division. The E. coli nucleoid appears as a clear region that excludes the dark staining ribosomes and contains many DNA strands, and that's over on the right. The nucleoid forms about 50 loops of chromosome that are called domains, that are shown as shaded areas. Okay, these radiate from the center, and then with each domain, the DNA is supercoiled and partially compacted by DNA binding proteins. And so those areas which are shown in green are not actively used in transcription in order to allow the bacteria to function under normal circumstances. Okay, so a little bit unique here. Once DNA has been replicated completely to form two daughter genomes, the cell may divide to form two daughter cells. Replication of the DNA termination site triggers the growth of the dividing partition of the envelope known as the septum, which grows inward from the sides of the cell, at last constricting and sealing off the two daughter cells, and this is called septation. Okay? So let's take a look at bacterial cell division, which is often referred to as binary fission.
Bacterial cells generally increase in numbers by the process of binary fission. The cell increases in length while doubling all of its parts, then forms a cross septum and divides to become 2 cells. 2 cells become 4. Those 4 become 8, and so on. This pattern of increase results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in numbers every 30 minutes. Other bacteria have generation times of several hours or even days. During growth of the cell, streptococci and some other gram-positive cocci synthesize new cell walls in the septal region. Rod-shaped bacteria such as Escherichia coli, Salmonella, and Bacillus species synthesize new cell walls over the entire surface of the cell.
So as you saw in the previous video, it's a fairly simple process. Under optimum growth conditions, it happens every 20 minutes. So bacteria go through a lot of generations very quickly. We have DNA replication, right? We have protein synthesis and cytoplasmic expansion, and the cell gets bigger, and then the septum forms and the cell divides, and then we have 2 new bacterial cells with, in theory, the exact same genetic information, unless there has been a mutation. Okay, and mutations happen, and sometimes their changes in the information are caught and corrected, and sometimes they are not. Okay, and mutation again isn't necessarily a bad thing, because if it produces a beneficial characteristic in the organism, it's more likely to establish a permanent niche in the environment. A niche is just a job that an organism does in an ecosystem in order for the ecosystem to function. Okay? Protein synthesis and expansion of the cytoplasm, septum formation and cell division, okay? And that process, known as septation, completes the binary fission process. Okay? Septum formation, which is septation, and motility determine the microscopic arrangement of cells. Cells from pairs have the prefix diplo in their name. If they're chains of cells, they have the prefix strepto, or if they're arranged like a chandelier, they are called palisades. Okay? Cells form packets of 4 or 8 when they septate in alternating parallel planes. Examples include cocci in tetrads and sarcinae. Okay? Remember that there are different shapes of bacteria. Based on those shapes, they have particular names. Rod-shaped bacteria are called bacillus. Okay, bacteria that are shaped like a coil are called spirochete, and bacteria that are round are called cocci. Okay, and I believe that's everything you need to know. So you'll get descriptions of bacteria like gram-negative rod, which would be purple rods under the microscope, or gram-positive spirochete, which would be red springs. Okay, so those are examples of designations. Okay. The packets of 4 to 8 represent the type of cell division that the bacteria undergo. Okay, whereas if they are going to form clusters, they get the prefix staphylo for cluster because they septate in random orientations. Okay, it's less well organized. We'll talk about what pili are and how stalks allow bacteria to attach to a substrate. We'll talk about flagellar motility and chemotaxis, and talk about thylakoids and storage granules and magnetosomes. Okay, so pili, as we stated earlier, are basically hollow rods that allow the transfer of genetic material from one prokaryote to another. Okay, so they're a little bit like a penis, okay, in that sense, in that they aid in the delivery of genetic material to another organism. Conjugation facilitates the DNA transfer between cells. Conjugation basically means to have a sexual method of reproduction. Okay, and stalks are membranous extensions of the cytoplasm that produce what are called adhesion factors, which are molecules that allow the bacterial cell to clip on and remain on a surface, like a tissue, for instance, and then to hopefully, from the bacteria's point of view, spread. Okay. Flagella are just mechanisms of locomotion for bacteria. Okay. You can have flagella in different orientations. You can have peritrichous, amphitrichous, and lophotrichous flagella. Okay, peritrichous flagella are all around the cell. Okay, lophotrichous look like horsetails at either end, and then there are um, um, basically amphitrichous, which are cells on uh, extensions, flagella extensions on one side of the cell or the other. Okay, and then you've got axial filaments in spirochetes that are similar. All right, they don't exist as cellular extensions, but they're kind of embedded on the surface of the cell, and they use ATP for movement. And when they do, the spirochete kind of rotates, and either he'll zip real quick to his next location, or he may do what's called tumble, where he starts to rotate in the opposite direction and remains basically in place. Okay, so those are just a few examples of locomotion. They all use ATP for power. Flagella is made up of a protein called flagellin, and you can see that there are other components to it. Right, you've got these rings here that anchor to the outer membrane and the cell wall, respectively. And then you've got these C rings that anchor under the cell membrane, and you're going to have other components that are actually embedded either within the cell membrane or between the cell wall and the cell membrane. Okay? The purpose of this whole setup is to basically spin that paddle around in either a clockwise or counterclockwise fashion, and that produces movements called runs and tumbles. Right, a run is where a bacteria zips in a straight line from one location to the other, and a tumble is where he stops, kind of fumbles around for a minute, and then changes the direction that he was going in. Okay, and that's determined again by interactions with chemicals in the environment outside the cell. Okay, bacterial cells tend to run towards useful components in the environment, like food and the proper pH and temperature, and that's called positive chemotaxis, and that they're also selected to run away from things that are toxic to them, and that's called negative chemotaxis. Okay? Multiple flagella are oriented in a bundle extending behind one pole, whereas their chemotactic receptors are at the opposite pole. When the cell veers away from the attractant, the receptors send a signal that allows one or more flagella to switch the rotation from counterclockwise to clockwise. And this switched rotation changes the bundle of flagella, causes the cell to tumble briefly, and then swim off in a new direction. Okay? So basically, it helps the cell get out of there. Okay? You can see some examples again of movement of these cells. Counterclockwise rotation of the flagella has proven the most effective mechanism for moving, for instance, pathogens from where they're found in nature to a willing host. Okay, it's one of the ways they get around. Okay. Other structures in bacteria: thylakoid membranes in photosynthetic bacteria, and then gas vesicles, which are found in aquatic bacteria and allow the bacteria to float at a particular level in the C column. Storage granules, where the cell will pack stuff that it needs later, such as glycogen, which can serve as a food storage molecule because it's basically polymers of a carbohydrate, which is basically designed to, in the case of particles of glycogen, store energy. Okay, in the case of gas vesicles, it allows the bacteria to float, Storage granules are areas where we can put stuff like glycogen or phosphate or sulfur, okay? And then magnetosomes can store iron oxides, and that allows the property known as magnetotaxis. This allows them to swing downward towards magnetic north, okay? We'll talk about the structure and the interconnections of the membranous organelles in the endomembrane system, and we'll describe the functions of the Golgi, the ER, and the nuclear membrane, and talk about the process of endosymbiosis that led to the mitochondria and to the chloroplasts.
Cells actually carry out life functions.
It contains the information, the unit of life.
Everything that is living is composed of either one cell, whether it be a bacteria or a protist, or multicellular.
Everything from humans to oak trees to fish and bacteria have cells.
The cell, the basic unit of life. The cell is the simplest component of living matter that can carry out all of the activities necessary for life in every organism. The simplest life forms are made up of only one cell, and in fact the vast majority of organisms are single-celled, like bacteria, amoebas, and paramecium. More complex life, like plants and animals, are multicellular, and these organisms can have trillions of cells. No one really knows the exact number of cells in the human body. It is estimated that there are anywhere from 10 trillion to 100 trillion cells, and about 200 different types of cells, with different shapes and various functions. While there is no such thing as a typical cell, most cells have chemical and structural features in common. The individual cell is actually a system unto itself, comprised of atoms and molecules, which combine to form proteins and various organelles which structure the cell. In multicellular organisms, like cells group together to form tissue, and tissues in turn combine to form organs, such as the heart. But the heart has many different types of cells to help it function. There are red blood cells which transport oxygen, white blood cells which help fight disease, and cardiac muscle cells that help the heart contract and pump blood throughout the body. Organs function together to make up organ systems, which ultimately make up an organism. And each cell's activity is dependent on the subcellular structures within the cell.
So a cell is not just something that we look at under a microscope and see, oh yeah, there's a cell. The cell is an activity, just as if you were to take a human city and speed it up tremendously with the way that all the people are moving around. So there's all this motion going on inside the cell.
The key to understanding how the body works, or how plants make food, as well as understanding the new frontiers like biotechnology and genetic engineering, is dependent on understanding cells. Most cells are microscopic. They cannot be seen without the aid of a microscope. But if you look at a slice of an orange, you will see it is actually made up of lots of tiny sacs. These sacs are large plant cells, visible to the naked eye. Cells are measured in micrometers and nanometers. A micrometer is 1/1000th of a meter, and a nanometer is 1/1000th of a micrometer. Because cells are so small, they remained hidden and unknown for centuries. It wasn't until the invention and development of microscopes that this miniature world was finally revealed, allowing scientists to study and begin to understand cellular life. In 1839, two German biologists, Matthias Schleiden and Theodor Schwann, developed an idea called cell theory. While looking at thin slices of animals and plants through a microscope, they saw how everything seemed to be made of tiny chambers. They called these chambers cells. Their theory states that all living organisms are composed of cells, and that cells are the building blocks of life. And while these were important revelations and are still accepted today, they still didn't know where cells came from. In 1850, Rudolf Virchow saw something that would change this. In his microscope, he observed cells pinching in two. He realized that cells actually come from pre-existing cells through cell division, and added this to the cell theory. Over the years, scientists have continued to develop this theory, and modern cell theory now states that all organisms are made up of cells, that new cells are always produced from pre-existing cells. The cell is a structural and functional unit of all living things. The cell contains hereditary information, which is passed on from cell to cell during cell division. And all cells are basically the same in chemical composition and metabolic activities. Cell biologists still cannot describe all the details of how cells produce the quality we call life. But recent research in genetics, developmental cell biology, immunology, and neurobiology have led to great strides in understanding structures, functions, and evolution of the cell. There are different types of cells, and certain cells have different functions, yet there are things all cells have in common, and one is a cell membrane that surrounds and encases the cell.
The cell has a membrane around it, which has various protein molecules on that membrane that serve as gates to let certain chemicals in and out of the cell. So the membrane is very important, kind of like the border of a nation is, with its gates to, let things, people or goods in or out of the nation. That's like the membrane of a cell.
It determines what substances are going to pass right through. It determines what substances need to have a messenger to take them through. It maintains the integrity of the cell. It contains all this material, all this organic material, the unit of life, from the outside world.
Within the cell membrane, there is a jelly-like substance called cytoplasm, which aids the structure and function of the cell. And while all cells function similarly, there are some important structural differences. Cell biologists group cells into two categories, prokaryotic and eukaryotic. Bacteria and archaea are prokaryotic cells. These represent the simplest, most primitive life on Earth, as prokaryotic cells are more simple than eukaryotic cells.
And that means that they don't really have a nucleus and they don't have a membrane that encloses the organelle. So everything is kind of, dispersed in the fluid of the cell, in the cytoplasm.
All other organisms like protists, fungi, plants, and animals are made up of eukaryotic cells, which are more modern and complex.
And one can even talk about the evolution of cells, the way cells have gotten more complex over time. Basically, our human cells are more complex than bacteria cells.
Eukaryotic cell has a true nucleus where the DNA is encased instead of floating around in the cytoplasm, and the rest of the organelles are encased by a membrane as well.
Organelles are special membrane-bound structures inside the cell which carry out specialized functions.
Just as we have organs within our body, the cell has organs within it. Because the cell is very small, these organs are called by cell biologists organelles. The animal cell has a very important organelle called the nucleus, which is where the DNA is housed.
DNA, deoxyribonucleic acid, is found in all cells. It is composed of molecules which carry genetic information. It is in a sense the instruction manual or blueprint for cells that specifies how proteins are assembled to regulate the cell.
What is it that DNA does? Is it the boss in the cell? Is it the kind of government in a cell? Not really. I look upon it more like the U.S. Constitution, in that it's it has to be actively gone to by the society itself and used to structure the government. That's for our society. For a cell, the DNA as a code is gone to by all the other molecules of the cell that are spinning around in their various interactions and metabolisms that are going to the DNA for the pattern of the code that makes the amino acids, that makes the proteins.
Chemical reactions and enzymes produce proteins which carry out the cell's activities, and ribosomes are where the proteins are made.
There are the ribosomes, small little bodies, and very, very many of them inside animal cells, and in fact, ribosomes are in all cells, and that is where the amino acids, small little building block molecules that make proteins, are brought together into chains, and these chains fold up into the proteins that are used, that all cells need, and that occurs on the ribosomes of the cell.
There's the endoplasmic reticulum, or ER, which is a network of membranes that encircles the nucleus and extends into the cytoplasm. The ER is responsible for synthesizing and assembling proteins and is where the ribosomes are located. There's the Golgi complex, a group of flattened sacs that are responsible for processing, sorting, and modifying the proteins, and the lysosomes, which are small sacs of digestive enzymes that break down ingested materials, secretions, and wastes. They are in a sense the stomach of the cell. Then there are the organelles called mitochondria. They are smaller than the nucleus, and there tend to be many of them throughout the cell.
And these mitochondria are the power plants of the cell. They convert the food that comes into the cell and combine it with oxygen to create energy molecules that power reactions in the rest of the cell.
Though plant cells are eukaryotic and have many of the same organelles as animal cells, such as a nucleus and endoplasmic reticulum, the plant cells have certain characteristics which are significantly different.
When we look at the plant cells, we see major differences with animal cells. Plants, of course, don't have skeletons. Each cell has its own skeleton, and that's the cell wall that go outside of the cell membrane. These cell walls are economically quite important. For instance, wood is all cell wall. Cotton fiber is a very pure kind of cell wall made up almost exclusively of cellulose.
But one of the most important differences distinguishing plants from animals can be found inside the cell. Plant cells have chloroplasts, organelles that contain a green pigment called chlorophyll, which traps light energy from the sun and combined with water and CO2 makes food for the plant through a process called photosynthesis.
And then there's one other structure that's extremely important that is largely unique to plant cells, and that's the vacuole. The vacuole is where water is stored. This ability to store water, to regulate water, is one of the things that makes plant cells be able to respond so rapidly to the environment. Very quick motions are centered around changing water distribution in the plants.
As cell theory states, all cells come from previously existing cells. This happens through cell division. Cell division, called mitosis, is an amazing and quite elegant process that enables organisms to grow and reproduce. And though there are some differences, this process is remarkably similar in all organisms. Through a sequence of steps, replicated genetic material of a parent cell is equally distributed to two daughter cells. It starts as all the chromosomes or genetic material are organized into entangled structures called chromatin. In the first phase, called prophase, the chromatin condenses into distinct chromosomes, and spindles that provide the mechanics for the division form at opposite poles of the cell. In the second phase, called metaphase, the chromosomes are aligned, equally distant from the two spindled poles. Next, in the anaphase, the paired chromosomes move to opposite ends of the cell. In the last phase, telophase, the chromosomes are cordoned off in distinct new nucleuses. This happens as cytokinesis divides the cytoplasm, making two distinct cells. Understanding the basics of cell structure and function is the foundation for understanding the future of science. Cell biologists, geneticists, and other scientists are investigating the properties of cells to find new ways to manipulate and replicate cells for use in agriculture, such as producing healthier and hardier plants. And in medicine, stem cell research is making strides in showing scientists how to make cells differentiate to do specific jobs, like increase insulin in diabetics, or possibly find cures for diseases such as Parkinson's or multiple sclerosis. Further investigation into how cells evolved and are continuing to evolve may uncover many of the yet unsolved mysteries of our world.
Okay, so as you saw with the eukaryotic cell, the architecture is a lot different than in something like a bacteria. There are membrane-bound and non-membrane-bound organelles whose purpose is to carry out a series of chemical reactions in a confined space that help the cell function more effectively, and to isolate chemical reactions that would oppose each other so that the cell can get effective amounts of work done. Okay. Other organelles are there for basically support and structure, and to provide a way for material to be moved around inside the cell or synthesized inside the cell. And we'll go through each organelle that you've already sort of seen a brief review of to give you a little bit more detail. One of the things that is critical in the way that a cell functions is it has to be able to manufacture the tools of life and then dump off those tools in the location in the cell where they're going to perform their normal function. And the way that that works is through something called the endomembrane system, in which the ribosomes, which are the builders of protein using information on messenger RNA, are able to manufacture those proteins on the rough endoplasmic reticulum. They end up inside the rough ER, travel through it just like a mouse traveling through a maze, and on the way towards the end of the maze, they fold into their normal shape, right? And then transport vesicles will move them from the rough ER to the Golgi apparatus, and inside the Golgi apparatus, they'll be chemically modified so that they can perform their normal job if that's necessary. So in some cases, they'll have sugars added to the proteins. In some cases, lipids will be added to the proteins. In some cases, phosphate groups or carbohydrates, or combinations of any of those. And then one of the chemical groups that's going to be added to the protein will be a chemical group that'll determine what secretory vesicle it gets loaded into, so that all the proteins destined for a particular location go into the same secretory vesicle and end up getting dropped off at the same location inside the cell. And that could be in the cell membrane, it could be to exocytose the material outside the cell, or it could be to to fuse with another organelle, another membrane-bound organelle, and drop off proteins either that are going to work inside that organelle or on the surface of that organelle. Okay, and all of this requires active transport. The vesicles that move and the pinching off of the secretory vesicles and all of that requires the use of energy in the form of ATP, as well as interaction with a component known as the cytoskeleton, which we'll get into in a bit. Now, the nucleus of the eukaryotic cell contains DNA, which is attached to proteins called histones, and together they make up something called chromatin. Okay, the DNA is the information molecule in the cell, as it is in the bacterial cell. It contains stretches of nucleotides that contain protein building instructions, and those are called genes. Okay, and so a gene is called on when the cell needs a particular protein made in order to fill one of its needs. Okay, the chromatin sits inside the nuclear membrane, and then when the cell is in a part of the cell cycle known as interphase, you will see a dark staining body called the nucleolus, which is the site of assembly of ribosomes. So what goes on in the nucleolus is that ribosomal DNA is transcribed into ribosomal RNA, and then ribosomal proteins are going to attach themselves to the ribosomal RNA and then become ribosomes, and then they will leave the nucleus through nuclear pores, which are basically tunnels in the nuclear membrane that let the ribosomes out so that they can go about doing their protein building work either in the cytoplasm or on the rough endoplasmic reticulum. Okay? The mitochondria and the chloroplasts that we find in single-celled organisms are that are eukaryotic are thought to be descended from ancient bacteria. Okay, and the reason for that is that when we look inside either a chloroplast or a ribosome, we find that it has its own DNA, and when we compare the sequence of that DNA to the sequence of bacterial DNA, we find a high degree of similarity. Okay, in addition, the ribosomes that bacteria use are functionally identical but structurally different from the ribosomes that are found in the rest of the eukaryotic cell. And it turns out that those bacteria-style ribosomes are found in both chloroplasts and in mitochondria. Okay? And we also find that the composition of the inner membrane of the mitochondria and the chloroplast is much more similar to the bacterial cell membrane than it is a eukaryotic cell membrane. Okay. The idea is that long ago, it's theorized that the early bacteria got together with some of the early eukaryotic cells and formed a relationship in which those bacteria would live inside those cells that benefited both partners, such that the bacteria got a place to live and a constant supply of food, and in turn the rest of the cell got the use of the energy produced by those bacteria, and the result was a relationship that benefited both participants, and that's called mutualism. Okay, a type of symbiosis. You can see here what the inside of a chloroplast and a mitochondria look like. The idea, again, behind the mitochondria is that it's the powerhouse of the cell. Okay, and I'm sure you've heard that maybe in high school biology. But basically, the chemical reactions that generate ATP go on inside the mitochondria, and that supplies the ATP for the rest of the cell. The reason for the extensive cristae in the inner mitochondrial membrane is to increase the surface area of the membrane so that you've got more workspace to generate ATP. Okay? Cells that use a lot of energy have more mitochondria in them, and the mitochondria tend to be bigger. Okay? And then if we look at a chloroplast, what we have are the grana, right? And then we have thylakoids, and again, these are little areas that are surrounded by membrane and populated with enzymes. And the reactions that go on here take carbon dioxide and water and turn them into carbohydrates. Okay, so the idea here is that a photosynthetic organism can make its own fuel utilizing the power of the sun, right? There's energy in the electromagnetic radiation from the sun, and the chloroplast uses that part of the visible spectrum, the red and the blue light, to manufacture sugars from CO2 and water, and then there are reactions that turn around and take those sugars and break them down in order to pull the energy out of those chemical bonds and transfer it to ATP. Okay, so that's why a green plant or a photosynthetic microbe is basically self-sufficient as long as it has light from the sun to allow it to continue to survive. Okay. The cytoskeleton is designed to provide shape to the cell and to allow the cell to move components around when and where it's necessary. Microfilaments are made up of a protein called actin, and what they do is they're important in processes like cytokinesis, and they are also critical in participating in some cases in cell junctions. All right, the microtubules are made of alpha and beta tubulin, and their job when the cell isn't dividing is to allow organelles to move along them from one location to another using ATP for power. And then during mitosis, the microtubules are important as part of the spindle apparatus, allowing the cell to basically divide its duplicated chromosomes between two new daughter cells. Okay, and then finally, intermediate filaments are designed to provide the cell structure, right, sort of a framework, and also serve in the context of tissues as part of certain cell-cell junctions like desmosomes and hemidesmosomes. Okay, microbes, you generally aren't going to see that too often, okay, unless we're talking about a multicellular organism. Okay, but you will see intermediate filaments providing a framework in single-celled organisms. Flagella and cilia are microtubule structures that are found in eukaryotes. Okay, the flagella in prokaryotes is made of flagellin, okay, and is structurally different but functionally similar to eukaryotic flagella. Okay, ATP is the power source. Okay, and the microtubules here are arranged in such a way that they can sort of ratchet over the top of each other using ATP for power, and then they sort of let go of that ratcheting movement, and then that causes the flagella to move back and forth, and that moves the organism from one location to another. Okay, contractile vacuoles are important in eukaryotes that live in a hypotonic environment where water tends to move into the cell as opposed to out of the cell, and if they didn't have a vacuole to take up and then eject the water, the cell would quickly swell up and rupture, just like a red blood cell would rupture in the process known as hemolysis. Okay, so this is a way for the cell to maintain its shape and its function even if it's in fresh water. Okay, where do you find vacuoles like this? Usually in protists. Okay. Some other animations that we will take a look at. You've seen active transport already. This is just another animation that will demonstrate it for you. And then bacterial cell division or binary fission you've already seen. This is just another video that will sort of walk you through that. Okay, so those will pop after this portion of the podcast is done. With that said, I will see everybody in the next podcast, which will be Chapter 5, and I thank you for listening.
cell division requires highly coordinated growth and expansion of all the cell's parts. Most bacteria have a circular chromosome that begins to replicate at its origin. At the origin, the DNA double helix begins to unzip, forming 2 replication forks. At each replication fork, DNA is synthesized by DNA polymerase molecules, which replicate the original DNA molecule, forming 2 copies. Replication proceeds in both directions around the circle. In a rod-shaped cell, the cell envelope and cell wall must elongate as well to maintain progeny of even girth and length. Growing bacteria replicate their DNA continuously with no resting phase. Sometimes even before replication terminates, 2 new replication forks form at each new origin of replication. Replication of the DNA termination site triggers growth of the dividing partition of the envelope, called the septum. The septum grows inward from the sides of the cell, at last constricting and sealing off the 2 daughter cells in a process called septation. Envelope extension and septation require rapid biosynthesis of all envelope components, including membranes and cell wall. Cocci, such as Staphylococcus aureus, do not elongate their cell walls before septation. Spherical cells expand their walls during septation. Here the furrows appear in the cell envelope all around the cell equator as a new cell wall grows inward. 2 new envelope partitions are complete. The 2 daughter cells peel apart. The facing halves of each cell contain entirely new cell wall. Transporting nutrients and wastes across the cell membrane often requires energy, using a form of transport called active transport. Most nutrients are present at lower concentrations outside the cell than inside, so the active transport further concentrates the nutrients inside the cell. Movement by active transport requires spending energy. In contrast, when a substance is transported from a higher concentration across the membrane to a lower concentration, the process does not require added energy. This is passive transport. In passive transport, a substance spontaneously moves down its concentration gradient. When a membrane protein provides a passageway, the passive transport process is more specifically called facilitated diffusion. Facilitated diffusion can move a molecule into or out of the cell. The net movement in one direction or the other depends on the direction of the concentration gradient. Consider that a concentration gradient represents a form of energy. As a substance moves across the membrane, down its concentration gradient, the energy of its movement can be harnessed to do additional work. For instance, it can fuel the active transport of another substance against its concentration gradient. The use of energy from one gradient to drive transport up another gradient is a type of active transport called coupled transport. When the 2 substances travel in the same direction across the membrane, the coupled transport is called symport. When 2 different substances are moved in opposite directions across the membrane, the active transport process is called antiport. In this example, the antiporter binds to substrate A on the cytoplasmic side of the membrane. As the antiporter opens to the outside, substrate A is released down its concentration gradient. Now substrate B can bind. The antiporter opens back to the inside of the cell, where substrate B is released, against its concentration gradient. Another way cells fuel active transport is by the use of ATP to move solutes uphill against their concentration gradients. The largest family of energy-driven transport systems are the ABC transporters, named for their ATP binding cassettes. There are 2 main types of ABC transporters, influx and efflux. Influx ABC transporters carry critical nutrients such as sugars, amino acids, and minerals. Efflux ABC transporters expel hazardous wastes and are generally used for removing antibiotics from the cell. An ABC transporter consists of 2 proteins that form a membrane channel and 2 cytoplasmic proteins that contain a conserved amino acid motif that binds ATP. An additional protein, called a substrate binding protein, initially binds the substrate, also called a solute. Substrate binding proteins have a high affinity for their matched solutes, so their use increases the efficiency of transport when concentrations of the solute are low. The substrate binding protein directs the solute to the channel protein. Hydrolysis of ATP to ADP plus phosphate yields energy to open the channel, allowing the solute to enter the cell. The transporter returns to its resting state.